All right, welcome back, everyone. More coverage of our cardiovascular system study today. We are going to be looking at the microanatomy of cardiac or of the heart, specifically cardiac muscle. So, in our previous lecture, we learned the path of blood. Just before that, we learned macroanatomy. We learned about the structure of the heart that you could see with the naked eye, and we learned about blood vessels, the great vessels entering and exiting the heart, and the difference in the different types of blood vessels. But today we're going much smaller. Today we're going to be looking at the tissue that makes up a heart. But as always, let's start with a not really attendant set of questions. First, what is a sarcomere? Second, where is calcium higher in concentration, inside or outside the cell? And third, where is calcium stored inside of a cell? Go ahead and try to answer those. Hopefully you've done that, so let's take a look to see what these are. First, what is a sarcomere? Remember, a sarcomere is the contractile unit of skeletal muscle. Today, we're going to see that it is also the contractile unit of cardiac muscle. Next, where is calcium higher in concentration, inside or outside the cell? Calcium is higher in concentration outside the cell, and actually inside the cell, there is very little to no calcium just floating around in the cytosol. And that's because, as we saw in Bio 137, calcium likes to bind to things. And when it does bind to things, it causes certain movements or activities, which maybe we don't necessarily want. In some cases we do. And where is calcium stored inside of a cell? Calcium, when it is inside of a cell, it is stored away in the smooth endoplasmic reticulum. So in skeletal muscle, they made use of this. They actually had a very modified smooth endoplasmic reticulum called a sarcoplasmic reticulum, and that was where it stored calcium until an action potential triggered that calcium to be released, and then that led to cross-bridge cycling. So all of this came from either chapter 3 when we studied the cell, or I believe it was chapter 9 when we studied skeletal muscle. If these are not clear to you, go back and refresh on them a little bit before we get into today's lecture, which is actually going to be a really short lecture. So, we're going to be studying the anatomy of of cardiac muscle cells, the muscle cells that make up the heart. And when we talk about muscle cells, we learned in Bio 137 that they are called myocytes. Myocytes. Now, when we're studying cardiac muscle cells, we can be more specific. We call them cardiomyocytes. Cardiomyocytes are the muscle cells that make up the heart. So cardiomyocytes, and we're going to be studying cardiomyocyte anatomy today. When we look at a cardiomyocyte under a microscope, it has some features that are similar to skeletal muscle as far as the way that it looks. It is striated. When we look at it under a microscope, it has those stripes just like we saw in skeletal muscle. And that is because it contracts using the sliding filament theory, just like we saw in skeletal muscle. Remember, the sarcomere had the different bands, the A bands, things like that, the I bands, the M line. Well, all of that is in cardiac muscle also. And when the cardiac muscle is contracting, those sarcomeres shorten due to the cross-bridge cycle. Now, unlike skeletal muscle, remember skeletal muscle was very long, very straight fibers, but cardiac muscle cells, they're very short and they branch. when we look at them in a microscope, they kind of look like a bunch of letter Y's laying on their side because of the way that they branch. So up here in the top left, this is cardiac muscle under the microscope. And here we can see that it branches. Right here we can see a branch. Down here is an artist rendition. So here we can see a cell coming down. It extends forward, but it also branches. And this is still that same cell that we see right here. It's branched. And since they are short and branched, well, one cell cannot wrap all the way around the heart. It does not reach that far. So instead, unlike skeletal muscle, cardiac muscle cells join end to end, and that's what we see right here. This is where one cardiac muscle cell ends and where the next one begins. They join end to end at a special feature called an intercalated disc. An intercalated disc is where one cardiac muscle cell ends and the next one begins, and they are joined together. So let's go back up here to the actual microscope image. We can see here a lot of very faint striations. These are those sarcomere striations, but here and there are some very obvious dark lines, and those are the intercalated disc. Here is an intercalated disc. It is where this cardiac muscle cell ends and this one begins. And we can see that this cardiac muscle cell, very short. Here is where the next cardiac muscle cell ends. And it looks a little hard to tell, but it looks like it's starting to branch right here. This looks like actually two intercalated discs just very close to each other. Now, an intercalated disc is a special type of cell junction. Think back to chapter 3 when we were learning about ways that cells are joined together. Well, two different types of cell junctions are found at an intercalated disc. We find gap junctions and desmosomes, and I'm going to talk about the desmosomes first. Desmosomes, another name for them is an anchoring junction. In this place, there is one cell has a special protein with a bunch of fibers sticking out. The other cell has a protein with a bunch of fibers sticking out, and those two sets of fibers grab onto each other, kind of like Velcro. This allows the two cells to hold onto each other very tightly, but it allows a little bit of wiggle room. It allows a little bit of motion. The cells are not going to go anywhere. They're holding on to each other very tightly, but it allows a little bit of motion. I like to think of this as if you've ever been out on the river on a boat and it's time to end the day. You pull the boat up to the dock, you throw the rope across, and you tie the rope around the little spot on the dock. Well, that boat's not going to go anywhere. It's tied tightly to the dock, but that boat is moving around a little bit. That's kind of what a desmosome is like. So desmosomes are there to hold the two cardiac muscle cells very tightly together. Now, over here, the gap junctions, also found at the intercalated disc, the gap junctions are like tunnels that pass from one cell into another. It is a pathway for things to move in between the joined cells. This allows the movement of ions through these gap junctions. Now, these are muscle cells, so they are electrically conductive. We have action potentials traveling along the sarcolemma, that plasma membrane of the muscle cells. But in addition to that action potential traveling along here, we also have ions passing very efficiently, very easy through from the cytoplasm of one cardiac muscle cell into the into the cytoplasm of the adjacent muscle cell, because those ions can pass through these gap junctions. And that allows for the action potential to spread through the entire heart very, very easily, very efficiently. And we're going to come back to that in just a few slides. But remember, these gap junctions, they allow the movement of ions between one cardiac muscle cell and the adjacent cardiac muscle cell. So let's look at some similarities and differences in cardiac muscle. Well, in cardiac muscle, some of the cardiac muscle cells are self-excitable. We'll see what that means in just a moment. When we were looking at skeletal muscle, remember skeletal muscle fibers had axon terminals from neurons attached to them. And those neurons sent an action potential to the skeletal muscle cell, and then that cell became excited, leading to a contraction. Well, cardiac muscle cells, some of these cells are self-excitable, meaning the heart does not require any input from nerves, from neurons, to cause it to contract. The heart, in effect, generates its own electricity. Now, the heart is all or none. Either the entire heart contracts or the entire heart does not contract. In skeletal muscle, remember we had motor units. So if you had one full muscle, for example, your biceps brachii in your arm, well, depending on which neurons are activated, we could have a few of the cells in the biceps brachii contract. We could have a few more. We could have a lot. We could have a lot more. We could have all of them. So just depending on the need of the muscle, we could have very little, very much, or all of it contract because of those motor units and recruitment. But the heart, we don't have that. Either the entire heart contracts or none of it contracts. Cardiac muscle cells have a much less elaborate sarcoplasmic reticulum compared to the sarcoplasmic reticulum of skeletal muscle. Remember, the sarcoplasmic reticulum of skeletal muscle was pretty large. It spread out over the sarcomeres very widely, and then we had those T-tubules where we had the sarcoplasmic reticulum expand into those terminal cisternae. Well, here, this dark blue area, this is the sarcoplasmic reticulum of cardiac muscle cell. We could see that it's almost looks like it's degraded. It's very thin. Well, we're going to see later that when cardiac muscle cell contracts, this plays a role because not very much calcium is stored in here. Yes, there's some, but most of the calcium, when we're talking cardiac muscle cell contraction, most of the calcium comes from outside the cell. So that's what we mean right here. Calcium from influx. Calcium, there is a little bit of it in the sarcoplasmic reticulum, but it doesn't play a whole lot of a role. Most of the calcium that is required for cardiac muscle cell comes into the cell from across the plasma membrane. Whereas in skeletal muscle, the calcium only came from the sarcoplasmic reticulum. In cardiac muscle cell, there is no chance of getting tetany, but in skeletal muscle, tetany is possible. So remember, tetany is when you contract a muscle and hold it in a contracted state. Well, that does not happen in cardiac muscle. In skeletal muscle, tetany is possible if you send a lot of action potentials very, very quickly, one after another, giving no time to relax. Well, in cardiac muscle, that's not possible because cardiac muscle has a very long absolute refractory period. Remember, the absolute refractory period is the time between one action potential and the ability to respond to another action potential. If you send an action potential through cardiac muscle cell, it must go through a full contraction relaxation before it will respond to another action potential. That prevents tetany. And that's a good thing. If we didn't have this, if your heart started beating very, very quickly, it would contract and hold itself in a contracted state. That is not something that we want. And lastly, cardiac muscle is almost entirely aerobic, meaning it needs oxygen. Skeletal muscle, we could have aerobic or anaerobic without oxygen. So in states where you're using more oxygen than you are supplying, skeletal muscle, it can withstand that. But cardiac muscle cannot. It uses a lot of oxygen because it is entirely aerobic. So, one last thing, and this really goes with our next lecture, but I want to go ahead and introduce the terms that we will be using in the next lecture. Now, if we were to take a heart and dissect it to where that we could see the inside, well, most of what we would see is cardiac muscle. Cardiac muscle forms the wall, and if we were to open those walls up, cardiac muscle runs throughout. Cardiac muscle separates the ventricles. Cardiac muscle forms a little tiny bit of the valves, but not much. The valves really are almost entirely endocardium. But cardiac muscle, when we look at the heart is almost entirely made of muscle. Now, the cardiomyocytes, the muscle cells, are the contractile parts of the heart. But there are some very, very specialized, very modified cardiomyocytes that do not contract. They have a few different names. We could call them non-contractile cardiomyocytes. Now, most cardiomyocytes are what we see right here. They are contractile cardiomyocytes. They contract and relax to cause the heart to beat and relax. But scattered throughout the heart, drawn here in yellow. Now, if this were a real heart, they would not be visually different from the rest of the heart. They are just drawn yellow here so that we could see them. These are still made of cardiomyocytes, but they are non-contractile cardiomyocytes. Another name for them are autorhythmicity cells, and that's going to tell us kind of what they do. These cells do not contract and relax. These cells generate electricity. Now, they are very similar to nerve cells, but they are not. They are cardiac muscle cells. They just do not contract and relax. Instead, they generate electricity. Now, there's going to be a few different areas of the heart that are given names, and we're going to learn those names, and we're going to see what do they do in our next lecture. Up here at the top of the right atrium, right next to the vena cava entrance, there is a patch of those non-contractile cardiac muscle cells. This patch is called the sinoatrial node, or the SA node. The SA node. This is also sometimes called the natural pacemaker of the heart. Next, at the bottom of the right atrium, right next to the tricuspid valve, there is something called the AV node, or the atrioventricular node. Coming off of the atrioventricular node is a string of those non-contractile cardiac muscle cells. This is called the bundle of His, HIS, or the atrioventricular bundle, the AV bundle. The AV bundle runs from the AV node into the first part of this wall that separates the ventricles. But it almost immediately splits into two bundles that run parallel to each other all the way down through this wall that separates the two ventricles, down towards the apex of the heart. These two parallel bundles are called the left and right bundle branches. Once they reach the apex, they are going to reflect upward through the walls of the ventricles. Now we can see a little bit of the branching here, but in reality, it branches all through the entire wall of the ventricles like a spider web. These are called Purkinje fibers, or the subendocardial conducting network. Now, what we are going to see in the next lecture is that this SA node generates electricity. It causes its own action potential. And that action potential, since it's embedded in other types of muscle cells, well, all muscle cells are electrically conductive. So when this guy generates an action potential, that action potential is going to spread across and down the walls of the atria. At some point, it's going to reach the AV node, who responds to that by generating his own action potential. And when he does that, that action potential is going to travel down the bundle of his, the left and right bundle branches, and up through the ventricle walls in the subendocardial conducting network, the Purkinje fibers. Once that happens, that action potential spreads through all of the regular cardiac muscle cells, or the contractile cardiac muscle cells in the walls of the ventricle. Why is that important? Well, one, this guy, however many times he generates an action potential, that's how many action potentials travel through the heart. If he generated, let's say, 75 action potentials in 1 minute, well, 75 action potentials would travel through the entire heart in a minute. If he generated 100, well, 100 action potentials would travel through the heart in a minute. And why is that important? Well, what happens to a muscle cell of any sort when it is given an action potential, when it is supplied with an electrical burst? What happens? It's going to contract and relax. So we call this SA node the natural pacemaker of the heart, because however many times in 1 minute he generates an action potential, the heart is going to contract and relax that many times in 1 minute. That is going to be the topics that we examine in the next lecture, where we are going to look at the electrical activity of the heart. We're going to be looking a lot at action potentials. We're going to be looking at different types of action potentials and how specifically those non-contractile cardiomyocytes actually work. So be sure to, before the next lecture, go back and review both action potentials and skeletal muscle contraction and the cross-bridge cycle. Those are going to be very important. All right. Thank you, and I will talk to you next time.